chapters 8 through 14, DNA and genetics. Okay, what's the difference between DNA and RNA? Well, what is not correct? What's not correct is D. So DNA, sugar, does not contain this O here, this oxygen, what's called deoxy. DNA replicates semi-conservatively. One is old, one strand is old, the template strand, and one strand is new. On the DNA strand, adenine always binds to thymine. Hydrogen bonds hold nitrogen bases together. DNA polymerase catalyzes the formation of two brand new strands of DNA, brings in the new nitrogen bases. Repair enzymes, it's easy. Recognize and snip out damaged DNA. Messenger RNA is shorter than DNA because the eukaryotic DNA contains introns that don't code for anything specifically. There are 20 amino acids. A codon is 3 base pairs long. And the 64 different codons is the genetic code. So the dogma, the central dogma of biology would be the correct order of information transfer would go from DNA to messenger RNA to ribosomes to proteins. Each codon specifies one amino acid. Mitosis and cytoplasmic division function in all of these are the functions of mitosis. Sister chromatids are attached at the centromere. Proteins that DNA wraps around are called histone proteins. Chromosomes are duplicated during the S portion of the cell cycle. The G1 stage is the interval within one set of DNA. So it goes G1S, G2. Okay, so mitosis, if a parent cell has 16 chromosomes, then each daughter cell will also have 16 chromosomes. The chromosomes are aligned with the spindle equator during metaphase. And then they start to move to the opposite poles during anaphase. The fact that sexual reproduction is energetically costly is not an advantage. That's an easy one there. Sexual reproduction results in new combinations of genetic traits. Alright, so what is not true? Sexual reproduction produces clones. That is not true. Alleles are different forms of the same gene. Through meiosis, let's see, all of these are true. And then the essence of meiosis is so that each gamete receives one member of each pair of homologous chromosomes. Meiosis typically results in the production of 4 haploid cells. Interphase is the longest phase of the cell cycle. This confused everybody here. Y'all were looking for stamens, because that's what you put in your study guide, right? The anthers were at it's the part of the flower that's at the top, and I probably confused y'all about that, because the stamens are the filament that hold. Well, the stamen is the whole thing, but but the anthers is what has the pollen and the sperm in it. So I know that was kind of confusing. Crossing over of non-sister chromatids occurs during prophase 1. Transfer RNA has the anticodon. Diploid organisms, all of the above is true on diploid organisms. Okay, so what's heterozygous? D is heterozygous. Both of those are heterozygous there. And how would you describe this? Heterozygous. If all the offspring of a monohybrid cross have big A, and you could do a little monohybrid cross, but what you end up with is the parents would have to be that too. Type O blood is going to be D. Homozygous recessive. People with blood type AB are, that's false, they're universal recipients. What's the phenotype if a person is homozygous dominant for skin texture? Their phenotype would be dry skin. Their genotype would be big S, big S. And as an autosomal recessive disorder will appear only if the parents both parents carry the gene. Okay, sex chromosomes, that's your X and Y. What's true is C, the X chromosome carries more genes than does the Y. Trisomy 21 describes Down syndrome. A karyotype is a photograph of an individual's chromosomes. Insertions and deletions, so the answer is C, cause frame shift mutations. Autosomes include all chromosomes except the sex chromosomes. And in humans, the 23rd pair is a sex chromosome. So on a Punnett square, the letters within the boxes represent offspring genotype, the probabilities. Gregor Mendel studied pea plants. Nobody ever misses that. Codominance describes human blood type. The letters outside the boxes of a Punnett square are the gamete genotypes. And then embryo cloning occurs in nature. Embryo cloning. That's just kind of an easy